Welcome to our final kingdoms video. This video is about Protista, and it's actually the one and only protist video that we'll have. So we only have a part one. You'll find pretty quickly that I am fascinated by this kingdom, especially because so many of them are parasites, and I think parasites are pretty interesting creatures. In the background here we have Giardia, which you're all probably pretty familiar with or have heard of. And these guys look pretty cool, I think, because they look like they have a little smiley face, like they're smiling back at you. Although some of them kind of have a frowny face a little bit, I guess. So kind of in general, and remember this is kind of back to that first day where we made that kingdom chart. So remember that Protista are eukaryotic. They're mostly unicellular and microscopic, but some of them are multicellular. We'll talk about some multicellular examples. They are autotrophic, heterotrophic, and both. Most of them reproduce asexually, but some can use conjugation, which we'll talk about, which is where they actually exchange some genetic material. It's not necessarily sexual reproduction because new individuals aren't produced, but we'll talk about that. And the cell wall may or may not be present. So what you might notice here is that it's kind of, they can be pretty much anything, and that's why they're referred to as taxonomic misfits, because they contain all eukaryotic cells. They can't be put into plants, animals, or fungus. So they're kind of our jumble kingdom, I guess. But they are pretty important evolutionarily because they're what we consider to be kind of the ancestors or the base of the three multicellular kingdoms that we have, the fungi, plants, and animals. So this image you're probably used to seeing, but these are remember our eubacteria and our archaebacteria, and then from that come our Protista, and then you'll notice that plants, animals, and fungi all kind of stem from Protista. We can divide them into three different groups. So we have our plant-like protists, or algae, animal-like protists, which are called protozoans, and our fungus-like protists, which are often referred to as slime molds. For the animal-like protists, these guys are autotrophic, and you can probably tell that just by looking at them, that they're green, so they're probably making their own food. They do contain cell walls, and most of them are immobile, though some of them can move, and we'll talk about some examples that can move. So some examples are red, green, and brown algae. Algae is multicellular if it's red and brown, but green algae can be either multicellular or unicellular. They are photosynthetic, so they are autotrophic. They make their own food, and they're not able to move. They live in different types of water habitats. So red algae is always in marine or salt water. Green algae can be in fresh marine, or it can actually also be in the soil. And then brown algae is always in marine environments. And they're classified by their pigments. So I've been talking about red, green, and brown. An example of brown algae that you're probably familiar with is kelp. And what's cool about them is that they are used in a lot of our foods. So they can be used in pudding or ice cream or marshmallows. Agar, which is what we used in the petri dishes for the bacteria lab, is also created from, is a product of algae. Diatoms are really cool looking. I think they're really beautiful. They are also unicellular. They are autotrophic, so they are also making their own food. They can't move, and they live in fresh or marine water, so another marine living protist. And their shells are made out of silica, which is glass-like, and that's why they look the way that they do. Then euglenoids, they are our plant-like protists that can move. Notice that this euglena has a flagellum, which allows it to move. So they're unicellular. They can be heterotrophic or autotrophic. And they can have one or two flagella. So this one only has one flagellum, but they can have two. And then they live in aquatic environments. An example is euglena, which is this guy right here. Then our animal-like protists, which we call protozoans, they are heterotrophic. They're all unicellular. They don't contain cell walls, and most of them can move, and they can move by cilia, which are hair-like projections, and we'll see an example of cilia close up. Flagella, which we've talked about, are a whip-like tail, and then a pseudopod, which is a false foot. So they'll kind of like amoebas, or these guys here will kind of flop their foot and then move along with it, almost like an inchworm, but a little more jelly-like. The protozoans, some examples are sarcodines, which are the amoebas. And again, they don't have a cell wall, which allows them to be more flexible. They are unicellular, heterotrophic, like all of them, and they move by a pseudopod. They live in fresh and salt water, and some of them can cause disease. So amoebic dysentery is an example of a amoeba parasite, or a kind of a condition that amoeba can cause, I guess. Another ones are flagellates, who can also be parasitic. They are unicellular, they're heterotrophic, and these move through flagella, which you might be able to guess based on their name. They can be either free-living or parasitic, and an example of a parasitic flagellate is trypanosoma, which causes African sleeping sickness. And then our ciliates are unicellular, like all of them, heterotrophic, like all of them, but they move by cilia, which are these small hair-like projections, and they can live in either freshwater or marine environments. This is a paramecium right here, but then we also have Vorticella and Stentor as examples, and you should pause this video and look up some images of these two. They look really cool. Sporozoans are non-motile protozoans, so they're unicellular like all of them, heterotrophic, but they can't move, and they are parasitic. So these are malaria, or the protozoan that causes malaria, which is Plasmodium, and there are I believe there are three different types of Plasmodium. One of them is Falciparum, which are these purple parasites that you see. Then we have our fungus-like protists. So these are heterotrophic decomposers. They contain cell walls. They're multicellular. They'll exist in different forms, and they make spores, which is, kind of makes them fungus-like, and they look a little bit like a fungus too. And that's how they reproduce. We have three types: water molds, downy mildews, and slime molds, but I'm going to combine the water molds and downy mildews. But we'll start with slime molds, which are kind of cute. So at least in this image, they are multicellular, they're heterotrophic, and they actually have amoeboid movement. So they kind of, the amoeba is the one that I was saying moves with that, kind of flops that pseudopod, and so we can describe movement in that way as well. And they hang out in cool, moist, shady places, which is true for all of our fungus-like protists. Watery downy molds are multicellular also. They're heterotrophic, but they are either parasites or they'll feed on dead organic matter. They also have amoeboid movement, and they also live in cool, moist, shady places. And a cool example of the watery downy molds is they're what caused the Irish potato famine that you probably learned about in history, that killed a very large number of people. Now I mentioned that our paramecium our protists can reproduce asexually, and here's an example with paramecia. So it's similar to the binary fission that we talked about with bacteria, where the organelles and the genetic information will replicate so that you have more of them, and then the paramecia will start to divide. There'll be a membrane that is placed in between the now two halves, and then they'll eventually split. Paramecium conjugation, however, is a little bit more complicated, and this is where they're exchanging genetic material, but they're not actually reproducing new individuals. So let's watch this animation first, and then we'll go into the details.
Paramecia are unicellular organisms with membrane-bound organelles. They undergo a process called conjugation, in which a pair of paramecia will exchange genetic information. This process is not considered sexual reproduction because new individuals are not formed. In the beginning phase of conjugation, two paramecia form a cytoplasmic bridge, and their diploid micronuclei undergo meiosis. After three of the newly formed haploid micronuclei dissolve, the remaining micronucleus undergoes mitosis. One micronucleus from each connected cell is exchanged. The two paramecia separate. The macronucleus disintegrates in each paramecium. The micronuclei combine and form a new diploid macronucleus. Each cell now contains a macronucleus, micronuclei, and a new combination of genetic information.
Great, so let's go through that step by step, and we'll talk about this more in class, especially if you have a bunch of questions. So one thing to mention is that and he talked about that in the animation, but the paramecia have both a macronucleus and a micronucleus, which serve different functions. One is for reproduction, and one is for kind of general function. So to begin with, the two individuals will line up, and they'll make contact with their oral groove, which is this little divot there, and that's what lets them actually exchange the information. The micronucleus, so this under here, will undergo meiosis, which will then form four micronuclei in each of the two cells. So it starts out as diploid, and after it goes through meiosis, it'll have four haploid cells. Then three of them will actually dissolve and will be digested by the cells, so you're left with one diploid cell. And then that diploid cell will divide again by, haploid cell will divide again by mitosis. And at the time that this has happened, you'll notice that the membrane between the two has disappeared or has been digested. So the two halves will then exchange one of their micronuclei. So I think about this as exchanging lunch with a friend, so that you share some of your lunch and you get some of your lunch from the friend, so you end up with kind of a combination lunch. So the cell membrane between the two halves gets rebuilt, so now it's been exchanged, and here we are where you have part of your friend's lunch, and your friend has part of your lunch. So the membrane between them will get rebuilt, and then the two cells will separate again. At this point, the two haploid micronuclei, so that we have the original one and then the new one that you got from your friend, will fuse into a single diploid micronucleus. So the original macronucleus, which was up here, has been digested by the cell's enzymes. But now we have this single diploid micronucleus. That will divide by mitosis to produce two identical diploid micronuclei in each individual. And that's not really shown in this image. They've kind of skipped that step. But one of the micronuclei will then become the real micronucleus, but the other one will grow to become the macronucleus. And again, they do have two different types of nuclei within their cells. Protists can be pretty ecologically important for different reasons. So they are the primary component of phytoplankton, which is a pretty important food source for many organisms. They carry out 70 to 80% of the world's photosynthesis. So even though they're so tiny, there's so many of them that they're generating a lot of food, and they're the base of most of our food chains. But they have some negative contributions as well. They cause disease, like malaria, sleeping sickness. We've talked about these. Responsible for the red tide, which you see here, which poisons shellfish. And then algae blooms result in fish kills, which you might remember from earth science. So I'm going to stop there, and for your professional paragraph for this video, I'd like you to summarize the process of conjugation that you just talked about in the past couple slides. So if you need to rewind and go back through that again, I went through that pretty fast, do that and write your professional summary on